Cell Biology
by Princess Hayes,
Kaela White,
Blessy Varghese.
On today's video, we will go into depth on the functions and structures of the animal cell, the plant cell, the bacteria cell, the nucleus, the endomembrane system, the cytoskeleton, and the mitochondrion. Animal cells are a type of eukaryotic cell enclosed by a plasma membrane, and it contains multiple organelles. The nucleus is a single rounded structure bounded by a double membrane that contains the cell's genetic information, and it is the location for RNA synthesis. The chromatin is an organelle within the nucleus made of DNA, RNA, and proteins that provide structure for chromosomes. The nucleolus is a distinct, dense, non-membrane bound spherical structure inside the nucleus made of proteins, DNA, and RNA, and it changes the RNA and combines it to proteins, creates and forms ribosomal subunits, and produces half of the cell's RNA. The endoplasmic reticulum, or ER, is a type of network made of tubules and cytoplasm with attached ribosomes that is involved in the synthesis of proteins and lipids. The rough ER is the part of the ER with attached ribosomes, and it synthesizes transmembrane proteins, interior organelle proteins, and secretion proteins. Smooth endoplasmic reticulum is the part of the ER without ribosomes, and it is the location for the biosynthesis of fatty acids and phospholipids. Ribosomes are tiny complex organelles made of RNA and proteins located in the cytoplasm and on the rough ER, and they are the site of protein synthesis where amino acids are built into polypeptides. The intermediate filament is one of the three cytoskeletal elements, and it is made of the polymers of proteins that are specific to different cell types that are formed in cable-like structures. They function to allow the cell to have mechanical strength. There are over 100 versions of intermediate filaments, and they can be found in various locations throughout the cell. The microfilament is the thinnest cytoskeletal element, and it is made of actin monomer polymers that form a helix. They also enable the movement of substances within the cell, and they reinforce the plasma membrane by crawling. Microfilaments help with the cell division in the process of cytokinesis, and it also helps with vesicle transport and muscle contractions. The microtubule is the largest cytoskeletal element, and it is a hollow tube polymers made of protein dimers. The protein dimers are combined alpha and beta tubulins. The microtubule also help maintain the cell shape and internal structure, and also enables movement of internal substances with the help of cilia and flagella. They also aid with cell division through chromosome segregation, vesicle transport, and organelle arrangement. The mitochondria is an organelle with a double membrane in which the inner membrane consists of folds, and it produces most of the ATP that serves as energy for the cell, and it is where biochemical processes of respiration occur. Lysosomes are organelles that have degradation enzymes surrounded by a membrane, and its function is to degrade macromolecules. The secretory vesicle are small organelles that secrete substances from the cell and transport cellular products. The peroxisomes are organelles inside the cytoplasm that contain reducing enzyme catalysts and usually some oxidases that break down specific organic molecules and synthesize other organic molecules. The vacuole is a small organelle that absorbs water and aids to turgor pressure. The pressure created by the water absorbance is also a form of support for the cell. They are also used for the storage of valuable nutrients for the cell's use. Centrosomes are an organelle near the nucleus that holds centrioles where spindle fibers are formed during cell division. The microtubule organizing center made of 2 centrioles, and it provides structural support. The plasma membrane is the external boundary of the cytoplasm that is made of proteins and lipids that regulate the flow of particles in and out the cytoplasm. The cytoplasm is a jelly-like filling made of mainly water and salts inside the cell that surrounds all of the other organelles. And the Golgi apparatus is a folded membrane and vesicles that modifies and sorts proteins and is involved with intracellular transport and secretion. It changes and organizes proteins and lipids. Those are all of the organelles of the animal cell.
Plant cell. Most of the organelles found in plant cell are also found in animal cell. So what are the differences that make plant cell different from animal cell? Firstly, the cell wall. Cell wall serves as a protective outside layer for the cell membrane, providing support and cell structure. It functions as a gate to determine what is allowed in and out of the cell. Next comes the chloroplast. Chloroplast is an organelle containing chlorophyll, which gives the greenness to a plant and produces energy. It enables photosynthesis process, converting sunlight into chemical energy. Chlorophyll is stashed in membranous sacs called thylakoids, which are found in the grana of the thylakoid membrane. Inside the thylakoid is lumen, and outside the lumen is the stroma. The next organelle is its vacuole. The central vacuole is a membrane-bound organelle containing water and other enzymes that function during the life of the plant. Structurally, the central vacuole is surrounded by phospholipid bilayer. It can digest food as well as expel waste products out of the plant cell. As you have noticed, plant cells consist of both chloroplasts and mitochondria. The chloroplast photosynthetic process of the plant is responsible for converting sunlight into chemical energy or food. While converting energy, oxygen is released from the chlorophyll. The oxygen is then utilized by mitochondria in order to create ATP for active transport and the release of minerals in the plant. Hence, it requires both the organelles for its functioning.
Before we begin to discuss the functions and the structures of the bacterial cell, what is a bacterial cell? When you think of bacteria, their role is notoriously known for causing diseases from tooth decay to massive destructions like the Black Plague. However, they are a beneficial species that is important for good health. Bacteria are prokaryotes, meaning they lack well-defined nuclei and membrane-bound organelles, as well as all chromosomes, or I should say nucleoid, since one of the most unique characteristics of a bacterial cell is that they do not compose of chromosomes, but are composed of single closed DNA circle. Another significant characteristic is that bacteria run small in size from relatively 0.5 to 10 micrometers, and has a large range of shapes it can come in. The smaller the size, the larger the surface area of the cytoplasmic membrane to volume, therefore the faster potential growth. Starting from the inside of the bacterial structure, there is the cytoplasm, that is a gel-like yet fluid substance that holds all of the other cellular components, such as the DNA, mesosomes, and so on. Within the cytoplasm, the mesh string in the middle of the cell is the DNA, which holds all of the cell's genetic materials. Compared to the eukaryotic ribosome, the prokaryotic ribosomes are smaller and have a slightly different shape and composition, and their main function is to build proteins by translating messages sent from DNA, similar concepts to eukaryotic ribosomes. The structure that appears to be attached to the plasma membrane is the mesosome, and it plays a role in cellular respiration and and increases the surface area of the cell membrane. This leads to the plasma membrane, which encloses the interior of the bacterium and regulates the flow of materials in and out of the cell. Surrounding the plasma membrane is the capsule that keeps the bacterium from drying out. The capsule is made up of polysaccharides. For extra measures of protection and regulation, that bacterial cell has the cell wall, which is the outermost wall. This gives the bacterium its shape, protecting it from the environment and protecting it from bursting. One of the last structures of the bacteria is the flagellum, which helps this bacterial cell to move towards nutrients. This is because the hair-like structures has propeller-like motions.
Nucleus. The nucleus is an important organelle because it coordinates the cell genes and regulates gene expression. It consists of a nuclear envelope, nuclear lamina, pores, heterochromatin and euchromatin, and spliceosomes. The nuclear envelope is a double membrane structure surrounding the nucleus. Its function is to regulate the movement of materials in and out of the cell and to provide structural support of the nucleus. Nuclear lamina. It is composed of intermediate filaments and membrane-associated proteins. Besides providing mechanical support, the nuclear lamina regulates important cellular events such as DNA replication and cell division. Nuclear pores. These are protein-based channels found in the nuclear envelope. They regulate the movement of molecules from the nucleus to the cytoplasm and vice versa. In most eukaryotic cells, the nucleus is enclosed by this nuclear membrane in order to separate it from the cytoplasm. Heterochromatin and euchromatin. These are two structural forms of DNA in the genome, which are found in the nucleus. Euchromatin is a loosely packed form of DNA found in the inner body of the nucleus, while heterochromatin is a tightly packed form of DNA found in the periphery of the nucleus. Around 90% of the human genome consists of euchromatin. The main difference between euchromatin and heterochromatin is that euchromatin consists of transcriptionally active regions of DNA, whereas heterochromatin consists of transcriptionally inactive parts of DNA. Spliceosomes. The initial transcript of DNA consists of non-coding regions or introns, which are spliced out with the help of spliceosomes.
The endomembrane system is a cellular system made of interconnecting membranes, and like some of the previously described structures, it contains a nucleus along with its nuclear pores and nuclear envelope, the smooth and rough endoplasmic reticulum along with its ribosomes, secretory vesicles, lysosomes, the Golgi apparatus, and a plasma membrane. Each membrane creates functional and structural divisions. All of the efforts of each organelle work towards editing, packaging, and moving lipids and proteins throughout the cell. The endomembrane system transports substances in and out of the cell without actually passing them through the cell membrane using vesicles through endocytosis and exocytosis. The rough ER makes the proteins and the smooth ER synthesizes lipids. Then the Golgi apparatus sorts and packs the substances delivered from the ER. Afterwards, vesicles carry the contents to other locations in the cell, including the plasma membrane and the lysosome. Lysosomes are made from combined transport vesicles from the Golgi system with endosomes, and they break down unnecessary components from the Golgi apparatus. The endomembrane system works to edit, package, and move lipids throughout the cell.
As previously described, the cytoskeleton is a frame that gives a cell its shape. As seen here, it is within the cell membrane. In a way, it is like the skeletons in a human body that holds the organs. The cytoskeleton helps hold all of the organelles. It also helps facilitate the movement of materials in and out of the cell. These functions are created based on three main components: active filaments, or you can say the microfilaments, intermediate filaments, and microtubules. Many scientists like to describe how the cytoskeleton, specifically the actin cytoskeleton, is involved with the endocytosis process. Endocytosis is a process where cells internalize non-particulate materials such as proteins or polysaccharides by engulfing them. Key factor here is because they are required for recycling of plasma membrane lipids and trafficking proteins. Research shows that actin cytoskeleton is related in a way that it traps endocytosis into restricted plasma membrane sites. How this process and filament relate is that the actin is regulated to allow the formation and movement of endocytic vesicles. Remember, microfilaments help with cell movement, although actin filaments function differently among different cells. For example, in a mammalian cell, the actin is a facilitator of the endocytosis process, whereas in a yeast cell, actin is part of the process of the plasma membrane deforming, causing invagination. Currently, researchers are trying to prove even more that cytoskeleton and endocytosis are related.
Mitochondria. Mitochondria is considered as a powerhouse of the cell. Mitochondria supplies adenosine triphosphate to the cells, which are used by the cells for various metabolic activities. ATP is manufactured through the process of cellular respiration, and most of the steps happen in the mitochondria. Mitochondria is a double membrane bound structure which consists of outer membrane and inner membrane. The multi-compartmental system of the mitochondria is useful for cellular respiration, allowing reactions to be kept separate and different concentrations of molecules to be maintained. In between the two membranes is the intramembranous space. The inner membrane is folded into cristae and the inner compartment is bound by mitochondrial matrix. The outer membrane displays eukaryotic proteins, whereas the inner membrane displays prokaryotic proteins. The matrix contains mitochondrial DNA and ribosomes. Evidence shows that endosymbiotic relationship of mitochondria shared with proeukaryotic cell was a result of DNA and ribosomes. According to the endosymbiotic theory, mitochondria and chloroplast were formed when a symbiont was engulfed by a proeukaryotic cell. In the symbiont relation of mitochondria, mitochondria took over the ATP production, and eventually the proeukaryotic cell lost the ability to produce ATP. In the endosymbiont relation of chloroplast used the light energy to reduce carbon through carbon fixation in order to produce reduced carbon compounds like glucose. Chloroplast also consists of two membranes, one eukaryotic in structure and the other prokaryotic. Like the mitochondria, chloroplast can self-replicate and has bacterial chromosomes and DNA.